University. He has done his PhD from Bharatidasan University. His research interests include discrimination of membrane proteins based on structure and functions, prediction of membrane, recognition of protein folds, prediction of secondary and tertiary structures, identification of important residues, stability, medium and long range interactions, development of thermodynamics. So note that Sir is from a physics background. He understands a lot of things. Okay. So mathematics, biology, and physics. So that's a great advantage, including that computer simulation also. He has written notable books. He has authored protein bioinformatics and professional books in lecture notes in artificial intelligence, lecture notes in artificial intelligence 2011. Okay. So, and he has, I think he has, so the list never ends. So he has more than 192 publications. Sir, this will take another half an hour, sir.
No, just a minute.
So really, we are thankful for giving your consent, sir. Again, apart from that, he has got many awards, starting from Junior Research Fellowship, so to Japanese scholarship, postdoc, and outstanding intellects, best paper awards. Okay, and of course, with all these credentials, definitely he will be editor for reputed journals. So he is an editor in open structural biology, BMC bioinformatics, current protein and peptide science, current computer biologia, open biotechnology journal. Okay, so I believe he has more credentials. What he has put up in his website is just a tip of iceberg. So from his expert talks, we will be able to gain a lot. Okay, gain a lot, get an insight. Okay, okay, that was about our expert, and I request our participants to listen to the lecture. So the lecture may be of 45 minutes or 50 minutes durations. Okay, after listening to the lecture, then we can open for a discussion. Okay, so I would like all the participants to adhere to the norms so that the lecture flows smoothly. Okay, so now I will hand over this session to our expert. Sir, please, sir.
Yeah, thanks, Mano, for the invitation and the introduction. So I will straight away start my talk because already actually he gave 1.5 hours and then reduced to 1 hour. Now finally ended up with 55 minutes. Okay, try to cover the topic in the given time. Okay, are you able to see my screen? Okay, good.
Yes, sir. Yes, sir. Yes, sir.
Okay. Okay, good afternoon, everyone. And this week we have a series of lectures, and I give 4 theory lectures and 2 online demos plus practicals. Okay. So my lectures are mainly based on the introduction to bioinformatics and databases, and the protein secondary structures, and protein sequence analysis, as well as protein 3D structures. Okay. So today I'll start with the bioinformatics as well as the database. Okay. So my lectures is usually not like just I'm talking, but usually it's a kind of discussion. This is what I usually prefer to do in the class. Okay. So what do you think about bioinformatics? How many of you are from biology or bioinformatics background? So participants, please feel free to talk or just write in the chat box. Both are fine. Okay. So what do you think about bioinformatics? Genetics and plant breeding, microbiology, Okay, so the participants are from different background, and yeah, so one of you mentioned about the computer analysis of biological information. Yes, okay. So here I show the basic aspect of bioinformatics. Okay, so if you see here, so I put the bioinformatics at the middle, and all other subjects so that are in the different circles. So it's a field of science in which the biology, computer science, information technology, everything into a single discipline, right? To understand or to analyze or to interpret the biological information using computers and statistical techniques, right? So we see here, so I put various disciplines, and the middle I put the bioinformatics. That means bioinformatics requires the information from different disciplines. It is not just a single discipline, right? If you talk about the bioinformatics, the first and foremost important field is the biology, because biology is a field or the subject of science, right, which produce experiments, right? Because if you cut into two pieces the bioinformatics, one side is biology and other side is informatics. So we need biological data, and we need the informatics to interpret the biological data, right? So without biological data, right, there is no advancements in the bioinformatics. So it first foremost importance is the biological experiments. Fine. So biological experiments, right? So can you tell me some of the biological experiments? What do you think of? Which type of data biological experiments can generate? Because the participants are from different backgrounds. So several of them you are from biology background, right? So what are the different types of data you can generate from biological experiments? You can start from the You can start from the DNA sequences. You can have sequencing. You will have the sequence data, right? And you have the structural information. You have tree structures. You have networks. You have expression profiles. You have pathways. So biology that can produce large amount of data. Recently, we can have the next-gen sequencing, right? So here we can have the sequencing of almost various genomes, right? Human genome they completed 10 years ago, right? So there are a lot of genomes that are available. So they can produce a lot of data. Now the issue is we have a lot of data available, and what to do with the data. Biologists can produce data, but how to interpret the data, how to understand the mechanism, right? Whether we have any general aspect on any of these systems, right? So for example, if you take a protein complexes, right? So biologists can study the specific system, right? They will understand that function or the mechanism of that specific system. But if you have a group of systems, for example, hundreds of protein complexes or protein nucleic acid complexes, is there any common mechanism to understand from these complexes, right? Here we need the informatics, right? So we need to analyze all the data what the biologists they produce, right? So now I have put different fields of science here, right? What is the contribution of mathematics and statistics? What they do? Because if we give the data, right, they will derive some equations. Okay, they can produce some equations and they try to relate some of the equations, for example, the multiple regression equations, to understand some of the aspects. For example, if you give the stability of your protein, right? So we have hundreds of proteins and we have stability data. And what aspects, what factors which determine the stability? So mathematics, usually they'll try to take some parameters and fit the data, right, and then to derive some equations. The next aspect is whether these equations are confident or these equations reliable or not. They do statistics. They do the p-values. They do the different types of statistical tests to see whether the data is significant or not. Fine. Okay. So if we talk about the physics, what is the contribution of physics in bioinformatics? Right. How the physics contributes to bioinformatics? Right, I'll go with the same example. For example, if you have a protein, right, so it forms stable 3D structures, right? What determines, right, the stability? What makes a protein stable? Because stable conformation is important for the function, right? In the physics point of view, we can analyze various interactions. So what is the contribution of electrostatic interactions? What is the contribution of van der Waals interactions? Or the hydrophobic interactions, right? So all these specific aspects, right, they can try to interpret. Okay, for example, a protein which is stable with the stability of minus 20 kilocalorie per mole. How do you get this number? right? It is a contribution from two major aspects. One is unfolded state and the folded state. But physics can explain what are the various contributions and how to get this number, right? The biologists can produce the data and we can interpret from the physics point of view, right? If we talk about the computing, right? What is the role of computing in bioinformatics? For example, you have hundreds of data, we have thousands of data, right? We have millions of data now, right? For example, we take the databases, we'll have millions of data. So it is required to analyze the data, fine, right? Analyze the mining. So we need programming. So we need to write programming, right? In this case, the computer science play a key role, right, to interpret the data. Then we take the chemistry, because I list of chemistry. Because in the case of small molecules, right, if you want to drug design, so we need the chemistry, right, to synthesize and to understand the for the drug design. And you put IT, information technology, right? So here it is required to develop databases, you're required to develop servers, right, so that everybody can access, right? In earlier years, we used to collect the data and we keep the data by ourselves because it is very difficult. So then if somebody requests, we send by post, right? They have to key in the data and it's very, very big mess. Now due to the advancements, right, in the high-speed internet as well as the high storage computers, it is possible to develop various servers and the databases, and easily you can communicate and equally you can share the data. Right now, the cloud computing and all these things are available, so you can share the data. Right. So this is the major aspects for the growth of bioinformatics. Right. One of the aspects is the high storage computers as well as you have the high internet connectivity. So in essence, I can tell the bioinformatics. It is about the acquiring or managing, analyzing, and understanding the data which the biologists can produce. Right. This is the major aspects of bioinformatics. Okay. So now what are the various aspects of bioinformatics? We think about bioinformatics, as we talk about bioinformatics, right? What are the various types of works? What various aspects it can deal about? Right? Here listed into 5 different aspects. First one is you can develop well-organized databases, right? I'll show you some of the databases in the next few minutes, right? So one of the major aspects, right? You can see you can develop the very well-organized databases. Can you list up some of the databases in bioinformatics? Right, GenBank, NCBI, right, UniProt, PDB, right, various aspects. So some of them I'll show in the next few minutes, right. So it's very organized databases because also they are available in the literature and they are accessible, and you can get very authentic information from all these databases, right. And the second aspect is if you have data, right, then the next aspect is we try to understand something or we need to extract some information, right. This is what I here mentioned about the hypothesis. So we can derive computationally, we can derive hypotheses, right? For example, we have some sequences, right? Or different groups of sequences. Some of them are DNA binding proteins, some of them are protein-protein complexes, right? We can try to derive some hypotheses. For example, if there is any domain which contains more number of positive charge residues, we can tell, okay, this could be probably DNA binding domain because it has high possibility of interacting with the DNA, there is negative charge. Right? So we can derive hypotheses if you have the data, right? This is the second aspect of bioinformatics. Okay, the third aspect, if you derive some hypotheses, then we'll try to develop some tools. Okay, we can use these features, and then you can derive some algorithms. Then we can derive some tools, and if you properly evaluate your tools, right? So then you can develop the servers, right? So that it can be used for the scientific community. Okay, and the fourth one, so I discussed about the virtual screening of compounds for drug development, right? So currently it is one of the most important topics. Okay, even the COVID-19, several groups, they are trying to develop drugs or vaccines or antibodies, right? So usually using this virtual screening, you can scan a very vast amount of libraries and you can try to identify some of the potential compounds. Okay, this could be the potential compounds for the drug development, right? Then the fifth one, I put the big data analysis because currently it is very boom in the field, right? Because first it started, bioinformatics started in 1980s, 1990s. At that time, there are many courses they started bioinformatics everywhere throughout the world. Then after some time, because it declined, and then again due to the advancement of the big data, now we need people to analyze the data, right? Because there are a lot of data available, right, using the next-gen sequences. Right, so we need to analyze the data. So these are the major aspects of bioinformatics, right? Okay, so we'll repeat. So can you tell what are the major aspects of bioinformatics now? So what are the major aspects of bioinformatics? Is databases, hypothesis, tools and servers, structure-based drug design, right? And then the big data analysis, right? So these are various aspects. I will explain a few aspects one by one. Okay, so I put an example here, right? Can you see my screen? Yes. Okay, I put 5 examples. Okay, this is the food pattern. Here is a food pattern. This is the control. This is yes or no. That means we know all the data, right? All experimentally known. This is just an example. Okay, they take the first example: 50% rice, 25% wheat, 10% meat, 10% fruits, and 5% vegetables not controlled. Right? Likewise, I give five different examples. Right? Now, if you can use these 5 examples, you can try to understand. You can try to derive some hypotheses. Right? What are the various factors? What are the contribution of different food patterns? Right? To become normal and to become not controlled or to become not controlled. Okay, so we use these hypotheses, then you can derive some equations. Okay, these are the probable equations, and then you can use the equation for the new cases. For example, if I give a test case, okay, this is your test case. So what is the answer for that? If one consumes 20% rice, 10% wheat, 10% meat, 20% fruits, and 40% vegetables, right? So what is the answer? This is controlled or not controlled? Controlled. So why it is controlled? What is the reason to derive this conclusion? Your answer is correct. So what is the, how did you get this number? What is the basis for this? Okay, vegetables 40%. Okay, but here here they have the vegetable 35%, but it's not controlled in the second case. Amount of vegetables and fruits. If you add up vegetables and fruit, it is 60. Yeah, that is fine. Yeah. Yeah, 55 is controlled. Okay, that is correct, right? Fine. So now we can, So we can prepare a model. Yes, you are right. It's controlled. So just we can give some examples. Okay, this is one example. Okay, this you can develop various models, right? So when you use this model and then use this equation for a new case, right? And then if you test various cases and everything is correct, then you can develop a model. Likewise, now this is the current scenario about the personalized medicine. If you go with any disease, they will ask about the history. Okay, so what is your blood sugar? What is about the blood pressure? Right? What is your pattern of food? And all these details they take, and your history they take, and they give you medicines. And finally, okay, they will derive some equations. Okay, if you have this type of symptoms, then you will end up with this type of disease, and these are the probable medicines. Okay, this is the current scenario is going on. But for this, we need patient data. That is the reason it is taking time. Right? Recently, several papers they published in Nature. So machines can do that rather than the doctors at the level of doctors level. If you provide the correct information, right, you can easily subscribe the probable medicine. This is the current scenario that is going on, right? So bioinformatics can handle large amount of data and they can provide the possible solutions for any cases, right? There is one example. This is another example. Okay, so here I put the 4 ponds and I put fishes here, right? So you want to catch fish. Where do you want to put your net? 1, 2, 3, or 4? 1, right? Of course, it is 1. If you take this 1, it is fine. If you put your net at the number 4 and you spend your whole time, you don't get anything, right? Likewise, if you have this disease, right, you can see the for suitable drug for any disease. You try with the compound 1, it won't work. 2, it won't work, and compound 3 could be probably a drug, right? So if you have this information, right? So currently there are a lot of data available in the literature. If you say all the compounds by trial and error, so there are 35 million compounds in zinc and 2.2 million enamine, and so natural compounds, Chinese medicines, and the Indian natural compounds and all. We cannot try one by one. If you try one by one, then by that time the patients will die. So we need to find someone, we ask someone, okay, what could be the probable ones? For example, if someone tells you, okay, you put your net in number 1, you will get fish. So you feel very happy because rather than wasting all the time in putting the net in 4, they get the number 1 and then they will get the fish. That is fine, right? So in this case, out of 35 million compounds, if one can tell or shortlist over 1,000 compounds or 500 compounds, then it is easy for the experimentalist. They can save time, they can save resources, right? And they can do the experiments, they can identify the probable compounds, right? So bioinformatics can handle large amount of data, and computer techniques assist to searching a drug target and designing a drug. Fine. So now this is one example we did. So on this aspect, okay, Tokyo Institute of Technology, they organized a competition, that's Initiative for Parallel Bioinformatics, IPAB, right? So what they did, so they gave 2.2 million compounds, right? And finally they asked us to give 120 compounds with order, with ranking, right? So they gave the target. The target structure is available, then we can take the PD from the structures. Not available, you have to do the modeling, right? Then we have these 2.2 million compounds. So how to do it to reduce to 120, right? So we did some computer strategy, right? So we used the computer algorithm. I'll tell you in a few minutes, right? And finally we gave 120 compounds, and they tested for 50 compounds, and the 4 of them showed inhibition, and one is the probable hit, right? They found this is the first competition, second competition again they found 2 hits, right? The one group, likewise different computer groups, right? They showed different compounds, right? Then finally they got about 100. So rather than doing this 2.2 million, they easily do the experiments on the 100. They can get IC50 values, they can do all the biochemical assays, and they can do the animal studies, and easily they can go up, right? And then try to identify potential drugs. This is our criteria, what we did. Okay, maybe a little bit too much for the audience, but just I'll put it in a very simple way. First, we have 2 aspects. One is the target, that's a protein. And the other side, we have the ligands, for example, 2.2 million compounds. Take the protein, the structure is not known, so you model the structure. But if you model the structure, we get one static structure. So in this case, we'll get only one structure, but the proteins are moving molecules, they are not stationary. So we did the MD simulations, and we got about 7 sample structures, because we generated thousands of structures and clustered them together, and we picked up the random structures from each cluster, and finally we got 7 clusters. This is one side. So we have protein side, we have 7 structures. And the ligand side, we have 2.2 million compounds. We search the databases and literature and identify some of the known ligands which could have the potential the inhibitor activity with the target. So we got some positives and negatives, and we removed redundancy, and using that we obtained the physicochemical properties, and we screened first based on the properties with the deviations. So we reduced it about 5,000 compounds, and we know these decays, they are not inhibited activity. This for the control, we took that, and we did the screening, and finally we identified these 120 compounds based on the elimination of this 63519 and the inclusion of this 5213. Then we got it. This is how we finally ended up 120 compounds. Fine. So this is the drug design aspect. Then we go to the big data, right? So currently everybody is talking about big data, right? We have the data analysis, and there are a lot of offers everywhere throughout the world. It started earlier in the physicists. They produce the data in petabytes. Mainly the astronomers and high energy physicists, right? They collect the data. Right? Recently, the life sciences also entered into that group, because it can produce large amount of data. For example, high throughput genomics, next-gen sequencing, and different genomics, proteomics, metabolomics. A lot of data life sciences can also produce now. So they also joined, right, in that group of this big data. So there are various initiatives in the big data analysis. For example, NIH in US, so they started this BD2K, that's big data to knowledge, right? There are 12 centers that to share the research objects, right? They collect the data, and they share the data, or they store the data, as well as for computing purposes. That is one initiative. In the same way, in the UK, they started 100,000 genomics projects. First, they started 1,000 genome projects, right? That was over, right? In 2010, it was over. And then they started 5,000 projects, that's done. Now the UK is doing the 100,000 genomics project to get the genomics of different ethnic groups, different people, and then see what are similarities and the differences to understand the person's medicine. And then the second thing, how to analyze the genomic data. They try to do 1 million individuals, This is a combination from US Precision Medicine Initiative as well as the Beijing Economic Institute in China. Right? These are various initiatives. Now in India, so we opened this consortium called OSCAT. This open source consortium for computer drug discovery. Right? So here also they try to collect all the open sources, right? Database and online tools, right? So we have the consortium so that anyone can use it for any analysis and they would derive any hypothesis and they can use it for the further applications, right? Fine. So we have the various concept databases, hypotheses, and the online tools, and the drug design and the big data, right? So what are the various challenges in big data in biomedicine? Right? The major challenges are the creating analytical tools and solving the scientific puzzles and how to access the gigabytes of data, millions of gigabytes of data. And this is the major things, this overcome the barriers to access patients' health records because it is completely confidential. So we have to do this scripting, right, to get the access the patient's record. So in the databases, right, if you see every databases say they have a lot of data, but only some proportion of data are used. So in this case, they are trying to make a subset of data where most of the users can use the data for the further analysis. Then in this case, they can efficiently use it. Then the development side, they can have the high protein research and big data to healthcare. And this is why they, in the recent article in Nature, they mentioned about the requirement of bioinformatics scientists, right? Mainly statistics as well as life sciences to analyze the big data in biomedicine. Okay, this is complex in biology. If you take the bioinformatics, you can see the application everywhere, starting from the nucleotide sequences, and then the structures, gene expression, protein sequence, protein structure, protein interactions, cell signaling, tissues, organs, physiology of the organism. There are various applications in all the complexity of the biological system. Okay, simplest one, we take this nucleic acid, and then we go to the proteins and the signaling and right physiology and all, right? So the major aspects, as I discussed, the databases, tools, drug design. So we talk about database, right? So what do you think about database? So what do you think about database? Collection of data, right? So we have the data which are very scattered in the literature, right? So it is required to collect all the data because the biologists, they produce large amount of data, right? So it is necessary to collect the scattered data and then put in the proper order to form a database. And essentially, you can say it is organized collection of information in computable form, right? Okay, so for example, if you want to develop a database, what are the various aspects you think about? Right, currently several databases are available, right? If you want to develop a database, what are the various aspects you think about to develop a database? Collection of information. First, we need to identify the theme. Okay, on what aspects do you want to have data, right? Because there are various databases available in literature, that should not be completely redundant. So you need to identify the area where you need the database and whether this area is important or not, and whether you will get the sufficient data or not. Okay, this is the first aspect you need to do it. And okay, now you okay. For example, if I tell okay, you define the theme, then what are the very other aspects you need to think about? Okay, we need got to see the correct structure. Yes. So second thing is you need to go authentic information, because if you develop a database, you should give any authentic information, because if you Google it for any aspects, also you receive a lot of WhatsApp messages, right? But if the messages or if the information is non-authentic, then you don't trust the data after that. So you have to give the authentic information and reliable information in your database, right? And the third aspect is, so data set should be available, right? For example, if you Google it, and one search takes 5 minutes. Do you use a Google again? If it takes long time? No, because if you know, nobody can wait for more than a fraction of a second. So if you Google it immediately next second, you expect to get the data, right? So when you develop a database, your database should be available all the time. It should be fast enough, right? And then what other aspects? Yeah, then another aspect is sometimes if you Google for any information, will you get the required information in the first line? Or sometimes you need to go for the further refine the searches or go to next pages. Right? If you develop a database, you should define okay, what the people or what are the users expect from the database. Right? So they should be able to get the information what they need. That you need to think about. Then if you give the information directly in your data search in your display options, right? Then the users will come again and again to use this data. For example, if you take Uniprot or the protein data bank or the GenBank, right? So we know that okay, this is search option we give, so you'll get the data, will get it, right? Fine. So these are the various aspects you think about. What are the characteristics of database? What are the various aspects we need to think about, right? Developing database. First, you need to have the contents. You have to decide what are the various contents we need to key in your database. And second, in your database, you use various technical terms. These technical terms are known to you, but not to all the users. So for all the technical terms, you have to list a valid terms and conditions, your terms and definitions, right? You have to show the definition. If you use EC number, you have to tell the EC number means enzyme commission number. If you put the PDB, right, you have to expand it protein data bank, right? Likewise, for each terms, you have to give the definitions. Then you need to have the logical structure. Okay, so what do you want to keep in? What are various data you will put inside your database, and what are the various links you have to provide, and all the information you have to derive. That is called the logical structure. That is called a schema, right? That's very important. I'll show you some examples, right? Then you have to have a format. So if you have a database and all the records, you should follow a specific format. For example, if you take the Uniprot or if you take the protein data bank, they use some specific format. Okay, right? First one is the compound and the header, protein sequence, structures and coordinates and so on, right? So they have a specific format. Then in fifth one is the retrieving the data. How to retrieve the data, right? You have search options and we can have the display options and they can retrieve the data for your search. Then next option is the links for information, right? Other resources, right? Related to the your database, and finally the backgrounds and so on. Okay, I'll show you the example here. Okay, so here this is your contents, right? So this is the thermodynamic database for the proteins and mutants, and here I give the protein name, source, PDB ID, and the number of molecules, secondary structure, and so on. These are contents, and the second one is ontology. So here for all the technical terms, what I show in the left hand side here, and all the technical terms, I give the details. Okay, here we provide the details for the explanations. Then we give schema. So here is our database called the Protham, and these are the experimental conditions we use, and the experimental data we provide in this, and these are the other links where we get the data from sequence data, structure data, mutation data, and so on. Right? And once we have it, and we have search and display options to analyze the data and visualizing the structures. Right? We can use it for the interface, and then we can use it for the analysis or developing the different models. Right? Then this is the format. We use some specific format, and if you see all the data, all the entries have the same format. We follow the same format, so that it will be easy for the users to fetch the data. Right? Then this is your retrieval. You have search options. You can search with various options, right? Whatever the users like to get the data, right? And you will get the results, right? Whatever they wish, and the results are linked with the original source, like the PubMed is here, right? And it will go to the original site, and you will get the more details about the complete data which are in the database, right? Right. So this is the various characteristics of a database, and once you are developing database, you need to think about all these aspects, right? To create a reliable and useful database for any topic of interest. Right? Fine. So this I skip. Okay. So when you talk about the database collections, right? So the journal Nucleic Acid Research, right? So they publish the first issue, that is called database issue, in every year, right? So for the biological databases. So okay, this is the journal website, and here is the databases website. and you can get the list of databases, right, which are published in the nucleic acid research, or you can also submit your abstracts if you publish in somewhere else. So it has a complete collection of databases, right, for the biological aspects. Okay, these are the different categories of databases in the nucleic acid research, right? So you have nucleotide sequence databases and RNA sequences, protein sequence, protein structures, thermodynamics, genomics, right, and the genes, diseases, microarray data, right, and the other molecular biology databases, literature databases, plant database, right. It's a big list. Okay, I showed you only some sample list here. So it has a large list of databases, and you have a subset of databases available in the website of the nucleic acid research, right. You can go through it, the website, and then this website, and you can get all the information regarding databases, right, database collections in nucleic acid research. Okay, so here we talk about the nucleic acid databases, right. I'll briefly cover the nucleic acid databases and the protein databases, right. If we talk about the DNA nucleic acid databases, right. So we have the DNA and RNA, right. So what is the DNA? Right, this is the one which carries the genetic information, right, and this is made and resides in the nucleus of living cells, right. So here you can get the names. The DNA, it contains a phosphate group here, and the sugar here, and the nitrogenous base, right. And there are 4 different bases for the DNA, like the adenine, cytosine, guanine, and thymine, A, T, C, G, right. So here is A, here is G. These are called the purines, and here C and T, right. These are the pyrimids. This is for RNA. So RNA, instead of thymine, you have the uracil, right. This is for the RNA. So we have the 4 bases, right, for the DNA and 4 bases for the RNA. Fine. So now we have various databases for the nucleic acid sequences, right. So the major databases for the nucleic acids, right, one is the DDBJ, the DNA Data Bank of Japan, and the EMBL, that is European Molecular Biology Laboratory, and the GenBank, that is maintained in the US, right. So initially they started the databases individually, and finally they made an agreement. So they exchange sequences via the SciNet 3 computer network. So and they link each other, and they can share the sequences so that they can maintain the databases all the 3 places, right? And easily you can access the data. So if you access any of the sites, you get the information regarding the DNA sequences, right? Then in addition, they have some different types of search options, and they have different types of analysis options, so that you can use any specific site of your convenience, and you can use the options available in these databases, right, to get additional information. Okay, due to limitation of time, I will just go through the slides very fast. But the evening, 3:30 to 5, we have the practical session. At that time, we will see more details about the contents of these databases. Okay, so this is the DNA Data Bank of Japan. You can use this website to access this database, and they have various options to search and analysis, statistics, submissions, and so on. So if you go to the analysis part, so this is the growth. Currently, we have the 6.6 trillion nucleotides and 2.4 billion sequences. So we have various search options. You can search with the different keywords. So they have the quick search, or they have the advanced search here, and you can search it. So these are the various contents. They have the name, source, section numbers, translated protein sequences, bases, and so on. Right. So here is your sequence. So finally, you can get the sequence here. Right. So here, how many A, how many T, how many C, and how many G? 80A, 74C, 82G, and 76T. What is the AT content here? For example, if I give this one and ask you what is AT content? AT content means how many A plus how many T divided by total number of nucleotides. So in this case, AT means this 80 plus 76, that is 156 divided by the total number of nucleotides. If you divide total number of nucleotides, that much you will get the AT content of that particular sequence. Fine. So we have advanced search. You have various options to use this database, right? If you search it, you will get the results, right? Then if you, these are the information available. So this is your final sequence for your specific search. And you can submit your data in the DDBJ, in the mass submission system, and they'll check it and they'll update the database. This one database called the DDBJ. And the second one is a GenBank. This is mainly an NIH, right? It's a sequence database. This annotated collection of the all publicly available DNA sequences, right? This is the GenBank maintained in NIH. So this is the contents. You can see the contents are more similar, only few differences, right? Maybe in the practical session you can see the similarities and differences in the different contents, or you can after the lecture you can go access these sites and you can see the similarities and differences, right, in these three different databases. So they have the publications and finally they have the sequences. We see here they have a sequence, but they are not listing about the ATCG here. But in the previous ones, you can see they listed up the ATCG here, right? Fine. So this is the GenBank, okay, various aspects, right? And you can search the GenBank, and when these are your links, finally you get the final information regarding your data. So we have DDBJ, and you have GenBank, and the third aspect is the EMBL. This is the nucleotide sequence database, right? So this is the recently they changed to ENA. That is European Nucleotide Archive, right? So you can have the direct submission here, and you can retrieve any sequences from this archive. This is your growth, and these are contents. You can see the similar cases here. This is sequence is 185 base pairs and 609A, 314C, 355G, and 581T, right? So you can easily see the information here. Okay, it has a search times. Okay, you can search and finally you get the results, right? Due to the time limitations, I don't go through details. You can do that. This is the databases for the DNA sequences. Okay, what are the different databases for DNA sequences? Can you list the sequences, the databases? DDBJ, EMBL, another one, GenBank, right? You can do that, right? Fine. These are DNA databases. Now, if you go to literature databases, there are various databases for literature. For example, chemical abstract, physical abstract, PubMed, and so on. And the biologists generally use the PubMed. It is freely available database, and we updates every day. We can see the lot of updates. So there is freely available database, right? I think most of you are familiar with the database, so I don't go through details. Just I will tell you. Okay, we search with the any keyword. Just give the keyword here, and you will get all the information, right, regarding that keyword, right? And you have options to have a summary or number of terms per page to sort out, right? And finally, you get the complete data here, right? These are DDBJ, and if you want to get the full text, just you go to the click, right? Okay, this is the website here. Now you can just Google it and get it the PubMed, and you open access. What's the meaning of open access? Right, there are two different options. One option is the others should pay for the charges, right? Because they have to maintain the journals. In this case, then the journal paper is available to everyone. So everybody readers it's free. Second option is so others don't have to pay anything. So in this case, journals ask for money for subscription, right? So if you want to see the journal, they have to pay to read the full article. There are two different options. Open access is open to all the public, but the expenses are paid by the others or the other institutions. So here you go to the full article, you go to the PDF, and then you can get this full data and so on, right? So then if you want to see related articles for any particular paper, you go to related articles, and these are the different articles, right? So there's various features. You can get the latest articles, you can get latest entries, and you can use any mesh terms and to get the retrieve any data what you like. I have a few practicals in the afternoon, so you can practice more on this aspect, right? Then we go to proteins. We have 10 more minutes. I'll finish up something. So what is the building block of proteins? Right. What is the building blocks of proteins? It's amino acids. So we have an amine group here, one carboxyl group here. So this combined this is amino acids, right? So there are 20 different types of amino acids for proteins. They joined together by the elimination of water molecule to get the any protein sequence, right? So these are the 20 different amino acids, right? So this is your full name and 3-letter codes and the single-letter code, right? So broadly they're classified into hydrophobic and hydrophilic residues. And the yes, here another classification called essential amino acids. Fine. So here these are hydrophobic residues. They are grouped aliphatic, aromatic, and sulfur containing. And the hydrophilic residues, so negative charge, positive charge, and the polar residues, they are responsible mainly for the hydrogen bonds or the electrostatic interactions and so on. Right, there are various databases for protein sequences. Right, first they started with the PIR, that is Protein Information Resource, in the Georgetown University. Right, in the meantime, the Swiss Prot they did in the Swiss Institute of Informatics. After that, they merged together and formed this UniProt, that is called the Universal Protein Knowledge Base. It is the currently available database for the protein sequences, UniProt, right, and it has all the information. It is very integrated database with all the authentic information on protein sequences as well as the protein functions. Okay, this I skip. Okay, in the UniProt, right, why it is very important? Because UniProt has the high level of annotation, right, and the minimum level of data redundancy. Annotation means you can see the description of the functions and domain structures for post-structural modifications and the variants and so on. And minimum level of redundancy and high level of integration with other databases. If you have the UniProt, it is integrated with various other databases, right? Like the string database or the protein data bank, and they have the pathways, and they have these nucleus, the complexes. So they have various databases. They link with the other database. They have the high level of integration with the database. So currently it has computer annotated 178.7 million sequences, and they manually curated 0.5 million sequences, right? So then totally about 180 million sequences, right, in the UniProt database, right? This is your statistics as of yesterday. We have 189 million sequences, right? Only 88.9 million sequences as of October 2nd, 2020. Then if you see more details, we go to the UniProt. This is the length distribution, and the average of about 300 amino acids length. And this is the amino acid distribution in the UniProt database. And if you see, some amino acids are highly frequent. For example, if you see, can you tell me from this graph which amino acids are frequent? Like this alanine and leucine and valine, glycine, these residues are highly frequent, and some residues are very rare. Can you see which residues are rarely occurring amino acids? Good. So which residues are rarely occurring amino acids? Cysteine, tryptophan, right? These residues are very rarely occurring amino acids, right? Because hydrophobic residues and the Charlie residues are very important because form the hydrophobic core for the initial protein folding, right? And they maintain the electrostatic as well as hydrogen bonds to keep the protein in a compact shape for the function, right? So there are various reasons for the specific combination of these residues in a protein sequence. Okay, it has various contents, right? So it has the name and origin of the protein, functions, localization, and post-structural modifications, amino acid sequences, and the details. Right, because the limitation of time, I don't go into details. Just you go through the details when you have time, or we'll see in this practical session. Right. So you have various functions, and you have the localization, right, and you have the post-structural modifications, right, and this is your structure, known structures in the protein data bank. And here you have the sequence. This is your sequence for the particular protein, right? These are modifications, right? And the various databases. So you can search the data for any names. For example, if I search the hemoglobin chain B, so you can get all the aspects, right? And also you can get the various sequence identity. You can reduce the identity, right, with 100%, 90%, 50% using the UniRef. Right? If you have 50%, this is your 50% sequence identity. This is the number of sequences with 50% sequence identity, right? So, and we get it here. This is the sequence in FASTA format. Fine. So this I skip now due to the lack of time. Fine. So now what can we do with the sequences? Because we have a lot of sequences, right? What to do with the sequence? We have DNA sequence, we have protein sequence, right? So now we can use it for derivation hypothesis. And you can try to understand the various structural and functional implications of these proteins. Right? For this, we need to derive various features. right? So everybody should derive some various features. So in order to facilitate, so to make the life easy, right? So we developed a software called the sequence feature that is from our group. So you can take the sequences, either the protein sequence or the DNA sequence, right? They can calculate various features. Just you can give your sequence and it will give you a lot of features as you want, right? So for example, if this is a sequence AIKLE, right? So you can take the average property. For example, this is the property A and AIL is 2 and K and E are minus. The values are this. You can calculate the average property, so add up these things divided by total sequence length is 5, so equal 0.4. This is one example. For in the case of the hydrophobicity, 20 different amino acids, they have 20 different values. So you can get the average hydrophobicity if you give just give the sequence, it can calculate the values for average hydrophobicity on that. This is one aspect, we will get the average values. Second aspect is you can get the mutation. What will happen if you mutate? If you change one residue to the other. For example, if you mutate isolation to leucine, okay, the value of isolation is 2, leucine is 2, the value is 20, net effect is 0. Right? This is what we do it in the protein side. The DNA side, we have the properties, we have the dinucleotide properties. So you cut into pieces, this AT, AG, cut into AT, TA, and AG, right? But sorry, is not this AG, right? So this is the numbers. So we can get the average and you'll get the numbers. So I'll show you the demo here now. This is your sequence. And if you want amino acid properties, so this will tell you the different properties like compressibility, hydrophobicity, polarity, and so on. Okay, the screen is not so clear. In the afternoon session, we can do in your computer. So in this case, it will be clear to you. So this is your values, your actual values, and as well as your normalized values. You get it here, and finally this way you can calculate for various of features like properties, substitution matrices, contacts, and so on. Right. Likewise, nucleic acid-based features. Right. So here you have the sequence here. Right. Either you can give the sequence, or you have the option to upload your sequence here. Right. So in the various options, we have the properties, we have the confirmatory properties, we have nucleotide content. So if you submit here, right, here's the data you'll get it. So you have the stacking energy, enthalpy, entropy, right? These are the averages property values here. You can get all the details, right? And here is your contents. Adenine content means what is the percentage of adenine, what's the percentage of cytosine, what's the percentage of GC, or the thymine, all the details you will get from our site. It's easy. Just you give a sequence and you click, you get the information. And using this information, you can derive some features and you can use it for your analysis or for the prediction. These are websites, so you can get it. So we have a lot of other databases in our lab, right? If you are interested, you can go through this website, right? Database website, we list a lot of databases. For example, thermodynamic database for the proteins and mutants, and we have the database for the membrane proteins, and we have a database for the protein aggregation, right? Summarized proteins, aggregation prone regions, and aggregation kinetics, as well as the structure of these aggregating proteins. We get all the information. Then we got databases for the protein interactions, right? So we have the Proximate for the protein-protein interactions and ProCaf for the protein carbohydrate interactions. We will get the binding affinity. So we can get more information. It's just published in 2020. Right. So we have the data for the disease-causing mutations. Like this is for the membrane proteins. This is for the all the mutations, right? So you can have these disease-causing mutations, and you can relate. So now we have stability data, we have affinity data, and we have disease data. So you can relate all this information. You can do a beautiful projects, right? Using by coupling all these factors, and you can do a lot of work by combining all this information. So I show some of these my books. Okay, you can read my books. Okay, protein bioinformatics I published in 2010 by Academic Press Elsevier, and interactions, right? So the World Scientific just published, right? You can use my books. It contains all the relevant information regarding this protein sequence and structure analysis as well as protein interactions, right? So I will just recapitulate once again. So what are the various aspects we discussed so far? I think I'm on time. So what is bioinformatics? Which field of science is very important for the growth of bioinformatics? Which will have science produce experiments, provide experimental data for bioinformatics? Biology, right? So biology is the most important one, followed by the other disciplines. Fine. What are the various aspects of bioinformatics? Database, guys. Tools, big data, drug design, and hypotheses, right? You can do a hypothesis, right? Fine. So what is a database? Collection data in a computer organized form, right? Okay. So what is the big data initiatives by India in India? I discussed about various initiatives, big data initiatives from US, right? And the 100,000 genomes, and they analyzing the data. And India, we have this OSCAD, that Open Resource Consortium, right, for analyzing the data. Fine. What are the various databases for nucleic acid sequences? GenBank, EMBL, DDBJ, right? Fine. So what is the database for protein sequences? How many data available in UniProt data now? What is the size? How many data available in UniProt? Approximately? $188 million sequence in UniProt database, right? Fine. So then what is the program we use to obtain the data features from sequence? I discussed one program, so we can obtain the features from sequence. That is sequence to feature, right? Sequence to feature tool we discussed. So there you can derive features, right, from the sequence to the features, right? Fine. So there are various server salts available that especially you can get, right? So that's available, right, to derive the features, right? Fine. So I think I'll stop here and then we will continue in the afternoon at 3:30. Okay. Okay. If you have any questions, yeah.
Participants can unmute their mic and ask questions if any.
Yeah, my most of the talks are available in the NPTEL. You can get my slides and you get my talks in the NPTEL. You'll get more details in that because here I provide a condensed version.
As Sir told, Sir is again available in NPTEL and Swayam courses portal. So all his slides as well as PDF versions of the lectures are available in the NPTEL portal. So you can go through that. Yeah, there are few questions. Chemistry, biology background can learn.
They're right. They're in comments. Yeah, that's good. Yeah.
They can learn. They can learn if they have the open mind and the will to learn. I think it's a highly interdisciplinary subject, bioinformatics. So it has biology, physics, chemistry, mathematics, and computer also is related.
Yeah, mathematics.
Sir, one question. I am from maths.
Yeah, mathematics background you can derive some because you can take any protein sequences or stabilities or structure or function, and you can derive some mathematical equations. Because if you see, there are various models they develop to understand the structural functions and the networks and all. Because now the mathematics is very used in network analysis, so you can derive these your equations to understand and to simplify the networks, and you can understand more about these structural functions. Yeah.
Yeah, there's a question from College of Engineering, Anway. So, you can follow. You can follow Sir's NPTEL lecture, and his book also is available.
Yeah.
So you can, yeah. Definitely, if you have the will to learn, you can learn.
Yeah.
Biotechnology is bioinformatics is highly interdisciplinary.
Yeah, if you see my lab, I have students from physics, students from chemistry, students from computer science, and students from biology. So almost all disciplines. From pharma, food technology, right? So if you put efforts, yes, you sure definitely you can achieve. Yeah. Okay, then see you at 3:30. So I will give you some of the questions. And I have one student from mechanical engineering. He is taking my course now. It depends upon your interest. Yeah. Okay, so I'll show some of the practicals. If you have time, just you go through these databases and the seek to feature tool, that websites, and we'll focus mainly. We give some questions and ask you to do it. If you're not able to do it, and we'll show how to do it. Okay? Yeah.
Yeah. On behalf of the participants and myself, coordinator, so I thank Professor for his first session. Sir, thank you very much. Yes, sir.
Yeah, okay,
And
good.
yes, sir. And one announcement for participants. So join us at 1:45 for the stress management class. So which will be handled by Ritika Pariwal. Um, okay, so please join us by 1:40 maybe. Okay, and also the afternoon session of our expert talk by 3:30. Yeah. Uh, yeah. Attendance will be shared. Don't worry. We will share you the attendance. Okay. You just follow the course. Okay. Take maximum benefit out of this course. Don't worry. We will give you the attendance. Okay. Yeah.
Okay, yeah, fine.
Yeah. Thank you. Thank you, participants.
Yeah.